Hello, this is part 2 of the Unit 1 Nervous System Review for Biology 30. So in this video, we'll talk about the brain and sensory organs, sensory nerves. The central nervous system is the brain and spinal cord. And in the brain, we have some vague kind of areas we need to keep track of. So we know that we have this region here-ish called the cortex. And the cortex is the outer folded region. We can see here this is a human one versus a rat one. We have more folds because we want to put more surface area into the same spot, where we have a lot of higher brain functions or just more acute other functions. So towards the front in the frontal lobe, we have a lot of our sense of personality, our sense of self, our sense of reason is in this frontal lobe. The thankfully no longer practiced lobotomy was an operation where they shoved a spike up your nose and just mushed it around in there so that you didn't have a personality anymore and you couldn't question what other people told you to do. The temporal lobe, by the temple, kind of by your ear, should be a good clue that we're talking here about hearing. So it is also related to memory and a bunch of other things as well, but hearing is what I want you to focus on. The parietal lobe, kind of up at the back here, related to touch perception, sort of things about how your body is and what it's doing. The brain stem we'll take a look at in this picture in a minute. The cerebellum, this region at the back here, is where we do balance, coordination, posture, sort of how your body knows where your body is. The brain stem, so the spinal cord leading up to the medulla oblongata and the pons, these older structures are related to more kind of primitive functions. Like the medulla oblongata is related to things like how fast your heart beats and how fast you're breathing. This is something that you don't have to have a very big brain to need to control. Everything that breathes and has a heart needs to be able to control them. So this is a pretty old structure. The pons acts kind of as a bridge between older and newer functions of the brain. In Unit 2, you'll talk a lot about the hypothalamus and the pituitary, so this region over here, but that's sort of an interface between your nervous system and your endocrine system. In your brain and spine, you have some gray matter and some white. The gray matter cells are the ones that don't have a neurolemma, which means they cannot fix themselves. You have a bunch of different types of sensory receptors. You have photoreceptors for light. You have chemoreceptors for chemicals. You have mechanoreceptors for physical things like heat flow or touch or pain. And we have thermoreceptors for like actual heat. These all do different jobs, but they all have one thing in common, which is they take something in the external environment and translate it into a nerve signal. When we look at a couple of specific structures in this course, we're going to focus on the eye and the ear. We can take a look at some of the structures involved and also how they work together. So for vision, we're using photoreceptors, and photoreceptors are all going to be back here on the retina of the eyeball. Your eye has lots of different parts. Very important, we have the sclera on the outside that keeps it all together. I try not to poke a hole in that. We have the lens, which is used to focus light that's coming in, pulled on by little muscles, and these ciliary muscles. We have the iris. So the iris is the colored part, in this case kind of bluey purpley. In most humans it's brown. And that can be opened or closed by those same ciliary muscles, exposing more of the pupil. The pupil is the window, the clear window back to the retina. It looks black back there just because there's no light source in your eyeball. The receptors are all on the back. The photoreceptors are all on the back of your retina. And there is an optic nerve, a cluster or a bundle of those nerves, those axons, that's going to head back to the occipital lobe in your brain. So we have all of these different regions of the eye, these different parts of the eye, and it's all working towards the idea of focusing an image on the back and sensing the light that's there. Is there a lot of light or not a lot of light? Just light is sensed by rod cells. They're sensitive to even low levels of light. You use those a lot in the nighttime. And the cone cells are good at color reception. And they are focused around one little area, the fovea centralis at the back of your eye, which is where almost all of your cone cells live. You've got rod cells more frequently. I know it seems like you can see color vision in your peripheral vision, but that's your brain kind of just pretending that for you. So the idea is you have some kind of image. It needs to get focused by the lens. The light is going to travel in a straight line, and that image is always going to show up upside down, but that's okay. Your brain is used to it. So when you see light, it's going to stimulate a cone cell or a rod cell, usually both, the photoreceptor. That's going to send a signal down the axon through the optic nerve, and it's going to go to the occipital lobe in your brain. That's right at the back of your head. Why is it at the back of your head? I don't know. It just is. When we're looking at the ear has two main functions. One, of course, hearing, and two is a type of proprioception, a type of sensing how your body is in its environment. So balance. So for the balance part, the semicircular canals are the ones that are going to be doing that job. These semicircular canals are oriented sort of like in three different axes, like an X, Y, and Z axis. And when you wiggle your head, when you spin, whatever, there are little hairs with little weights on the end of them called otoliths that can wiggle back and forth. And they're attached to a mechanoreceptor, which is a type of a nerve cell that's going to send a signal to your brain. And that's going to give you orientation information. Am I upside down? Am I falling? That type of thing. The hearing part is going to be mostly happening in here in the cochlea. Of course, the sound has to get there first. So sound waves are pressure waves, and those pressure waves are going to go through your auditory canal, assuming that it's not all plugged up with earwax. And it's going to reach your eardrum or tympanum. That's going to shake, and it's going to shake the three smallest bones in your body. And those three smallest bones in your body are going to shake another little window and go towards this snail, this cochlea. That is where you're going to sense different frequencies of sound. Inside of your cochlea, you have, similar to inside your semicircular canal, little strings that wiggle when there's pressure applied to them. And those are more formally labeled the organ of Corti. The organ of Corti will sense not only how loud the sound is, but also the frequency of the sound. You'll notice that your cochlea is wider at the start and narrower at the end. You're going to hear low frequencies here and higher frequencies there. So for a sound signal to get to your brain, it has to come through here, shake the tympanum, go across the ossicles, your three tiny bones there, shake your oval window, foramen ovalis, and it's going to send its signal through the cochlea, activating the organ of Corti, and that nerve signal is going to be sent down the auditory nerve to the temporal lobe in your brain. All of this I think you've heard before, and my advice is always going to be, when studying for an exam, do as many practice problems as possible. I hope this helped. If not, feel free to reach out. If anything wasn't clear, let me know, and I'll try to make a better version.